Imagine having the power to change the blueprint of life. DNA can now be rewritten. This opens doors to new medical treatments. Fasting can influence our DNA. It affects gene expression and cellular processes. Fasting triggers our body's maintenance systems. Our cells go into repair mode. This can change how our genes are expressed, activating genes that promote health and longevity. CRISPR-Cas9 is a groundbreaking technology. It allows scientists to edit genes with precision. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. Cas9 is an enzyme that acts like a pair of molecular scissors. Together, they can cut DNA at specific locations. The process begins with a guide RNA. This RNA is designed to match the DNA sequence that needs editing. The guide RNA leads Cas9 to the right spot in the genome. Once there, Cas9 makes a cut. This cut can delete, insert, or modify genes. CRISPR-Cas9 has many applications. It can be used to treat genetic disorders. For example, scientists have used it to correct mutations that cause diseases like cystic fibrosis and sickle cell anemia. It can also be used in agriculture to create crops that are more resistant to diseases and pests. Despite its potential, CRISPR-Cas9 raises ethical concerns. Editing the human genome can have unforeseen consequences. There is also the risk of off-target effects, where unintended parts of the genome are altered. As we advance, it is crucial to navigate these challenges responsibly. Gene therapy is a powerful tool in genetic engineering. It involves replacing, inactivating, or introducing genes to treat diseases. The goal is to fix genetic causes of disorders. One method is to replace a faulty gene with a healthy one. This can be done using viral vectors. Gene therapy has shown promise in treating various conditions. Despite challenges, its potential is immense. Epigenetics is the study of changes in gene expression that do not involve alterations to the DNA sequence. Epigenetic modifications can turn genes on or off. This happens through processes like DNA methylation and histone modification. DNA methylation involves adding a methyl group to DNA. This can prevent certain genes from being expressed. Histone modification alters the proteins around which DNA is wound. Changes to histones can make DNA more or less accessible for transcription. These modifications play a significant role in development and disease. For example, epigenetic changes can influence cancer progression. They can also affect conditions like obesity and mental health disorders. Unlike genetic mutations, epigenetic changes can be reversible, making them a target for therapeutic interventions. Epigenetics bridges nature and nurture. It shows how our environment and behaviors can impact gene expression. This field offers insights into how lifestyle factors, including diet and fasting, can influence our genes. Somatic versus germline editing. Understanding the difference. Genetic editing can be done on somatic and germline cells. Somatic cells are any cells except sperm and egg cells. Germline cells are the reproductive cells. Somatic editing targets only the individual. It does not get passed on to future generations. Germline editing involves heritable changes. This raises significant ethical concerns. The debate over germline editing is ongoing. Section 6. Fasting and its impact on gene expression. Fasting is not just about skipping meals. It influences our genes in profound ways. When we fast, our body initiates a process called autophagy. This is the cellular cleanup process. It removes damaged cells and regenerates new ones. Fasting also affects longevity genes. These genes, like SIRT1 and FOXO, promote cellular repair and stress resistance. They play a role in extending lifespan and improving health. DNA repair mechanisms are also activated during fasting. This helps maintain genetic stability and prevent mutations. Histone modification is another impact of fasting. It makes our DNA more accessible for gene expression. This can activate genes that are beneficial for health. Fasting also reduces inflammation. It modulates the expression of genes involved in inflammatory responses. The changes in gene expression during fasting have many benefits. They can improve metabolic health, enhance brain function, and reduce the risk of chronic diseases. Understanding these processes helps us see the connection between diet and genetics. Section 7. Key molecular pathways activated by fasting. Several key molecular pathways are activated during fasting. One of the main pathways is SIRT1. This gene is part of the sirtuin family, which regulates cellular health. SIRT1 activation promotes DNA repair, reduces inflammation, and enhances metabolic function. Another important pathway is AMPK, or AMP-activated protein kinase. AMPK is an energy sensor in cells. When activated, it helps maintain energy balance. It promotes autophagy, improves insulin sensitivity, and enhances mitochondrial function. FOXO proteins are also crucial in fasting. These transcription factors regulate genes involved in stress resistance, longevity, and metabolism. Activation of FOXO proteins enhances the body's ability to cope with oxidative stress and inflammation. These pathways work together to promote health and longevity. They show how fasting can trigger beneficial changes at the molecular level. By understanding these pathways, we can harness the power of fasting for better health outcomes. Section 8. Benefits of fasting on health. Fasting offers numerous health benefits. One of the key benefits is reduced oxidative stress. Oxidative stress is a major factor in aging and chronic diseases. Fasting enhances the body's antioxidant defenses, reducing damage from free radicals. Fasting also helps prevent age-related diseases. It improves insulin sensitivity, reducing the risk of type 2 diabetes. It enhances cardiovascular health by lowering blood pressure and cholesterol levels. Fasting can also improve brain function, reducing the risk of neurodegenerative diseases. Metabolic health is another area where fasting shines. It promotes fat burning and weight loss. It can improve lipid profiles and reduce inflammation. These effects help prevent metabolic syndrome, a cluster of conditions that increase the risk of heart disease, stroke, and diabetes. The benefits of fasting extend beyond physical health. It can improve mental clarity and focus. Fasting has been shown to enhance mood and reduce symptoms of depression and anxiety. These wide-ranging benefits make fasting a powerful tool for overall well-being. Section 9. Types of fasting and their epigenetic benefits. There are different types of fasting, each with unique benefits. Intermittent fasting involves cycling between periods of eating and fasting. Common methods include the 16-8 method, where you fast for 16 hours and eat during an 8-hour window. Intermittent fasting can improve insulin sensitivity and promote weight loss. Prolonged fasting involves fasting for longer periods, typically 24 to 72 hours. This type of fasting can enhance autophagy and promote cellular regeneration. It has been shown to reduce inflammation and improve immune function. The fasting mimicking diet, or FMD, is another approach. It involves eating small amounts of food that mimic the effects of fasting. This diet can promote cellular repair and reduce risk factors for aging and disease. Each type of fasting has unique epigenetic benefits. They can influence gene expression in ways that promote health and longevity. Understanding these benefits helps us choose the right fasting method for our goals. Section 10. Conclusion: the future of genetic engineering and fasting. The future of genetic engineering and fasting is promising. Techniques like CRISPR-Cas9 and gene therapy offer new ways to treat and prevent diseases. They allow us to rewrite the genetic code and improve health outcomes. Fasting, an age-old practice, also holds great potential. It can influence gene expression and activate beneficial molecular pathways. The combination of genetic engineering and fasting can revolutionize healthcare. It offers a holistic approach to health, integrating modern science with ancient wisdom. As we move forward, it is essential to consider the ethical implications. Balancing innovation with responsibility will be key. By understanding the science behind genetic engineering and fasting, we can make informed decisions for a healthier future.